Okay. Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bandettini, and here I interview neuroscientists and discuss their work, as well as the latest developments, issues, and controversies in the field of brain mapping. Today, I'll be talking with two esteemed leaders in their respective fields, one in fMRI methodology development, and one in electrophysiology and pharmacology and clinical translation. We'll be taking a critical look at fMRI, discussing its limits and potential, and hopefully providing some clarity on what fMRI is, isn't, and what it could be, and how it fits in the wider scheme of things. So my first guest is Dr. Dimitri Kullman. He's a professor of neurology at UCL Queen Square Institute of Neurology. He received his doctorate from the University of Oxford in 1984, and his bachelor in medicine and surgery from the University of London in 1986. He alternated between research in synaptic transmission and postgraduate medical training in London. In 1992, he started his lab at the Institute of Neurology. And in 2000, he became a fellow of the Royal College of Physicians, which he belonged to previously. His interests spanned the fundamental mechanisms of synaptic transmission, the computational properties of small neuronal circuits, and alterations in neuronal and circuit excitability in epilepsy and other neurologic disorders. The core methods of his lab are in vitro electrophysiology and pharmacology, but he also applies confocal and two-photon laser scanning microscopy, computational simulations, molecular genetic methods, and heterologous expression of mutated ion channels. His laboratory has contributed to the discovery of silent synapses, glutamate spillover, presynaptic GABA receptors in the cortex, human epilepsy caused by potassium and calcium channel mutations, tonic inhibition of the hippocampus, and Hebbian and anti-Hebbian LTP in hippocampal interneurons, or long-term potentiation hippocampal neurons. One of his goals is to understand how phenomena that he studied at the cellular level interact to regulate excitability of small neuronal circuits. He's integrating studies on hippocampal circuit function with knowledge of how interneurons and principal cells fire during different behaviors. This is being approached both experimentally and with simulations. He also aims to apply his lab's recent insights into cellular consequences of inherited mutation of ion channels. Channelopathies, or as they're called, to develop new ways to diagnose and treat neurological diseases. He was also the editor of the journal Brain from 2014 to 2020, or just recently he stepped down. He brings to the table a perspective of a clinician neuroscientist who does research at the neuronal scale. So my second guest, Dr. Vince Calhoun, is the director since 2019, I think, of translational research in neuroimaging and data science called Trends, which includes 3 universities, Georgia State, Georgia Tech, and Emory. In 2002, he received his PhD in electrical engineering from the University of Maryland, Baltimore County, then became an assistant clinical professor at Yale and director of the Medical Image Analysis Laboratory Institute of Living in Hartford. He moved on in 2006 to the University of New Mexico in Albuquerque, and starting as an associate professor, and then moved up to become distinguished professor at the University of New Mexico and a director in various forms of the Mind Research Network in Albuquerque. Vince is an expert on brain imaging acquisition and analysis and has created numerous algorithms for making sense of complex brain imaging data. He's the creator of group independent component analysis algorithm, which has become widely used for extracting networks of coherent activity from functional MRI data. He was also an early innovator in approaches to characterizing the dynamics of brain connectivity. He's also developed techniques to link many different types of data called data fusion, including various types of brain imaging, structural, functional connectivity with genomic and epigenomic data. A key focus of Calhoun's work is the development of tools to identify brain imaging markers to help reveal and potentially treat various brain disorders, including schizophrenia, bipolar disorder, autism, Alzheimer's disease, and many more. He's recently served as the president of the Organization for Human Brain Mapping. And of all the people in fMRI, I have to say he's been probably the most prolific in his publications and his work to push methodology and hopefully applications of fMRI. So in this podcast, we're going to talk, our focus is fMRI. And fMRI has been around for 30 years. And as most people in this audience know, it's been used in many ways, and it has many limits. You know, one of the main issues is the fact that it relies on hemodynamic responses, and people have used this technique very well for answering some very good hypothesis-driven questions. We've gone through many stages of both advancing the methodology and having, mild crises along the way. There's a reproducibility crisis. There's been the debacle with the dead salmon that just simply illustrated, if you don't do your corrections properly, you can get false positives, and other things like that. And this podcast is a discussion of what those issues actually are in reality, and also how they've been addressed and how fMRI can move forward in the future. And this podcast was partially precipitated by an editorial that Dr. Kullman wrote to be, purposely provocative, and it was provocative, but it actually brought up some good points that I think are worth talking about. And that is one of the main points is that fMRI is a indirect measure, but is that really a damning problem or not? And of brain activity, and it has low resolution, it has low temporal resolution, and so on. Does it limit us from getting at mechanisms of brain function? Does it limit its clinical applicability? And the second criticism has been that, a lot of times we rely on the statistical maps with fMRI, as opposed to looking at the effects themselves and estimating the effect sizes. So we'll talk a little bit about that as well. So just to begin, I'd like to maybe start with Dr. Kullman to maybe fill in where I didn't fill in properly, or just to express your perspective, maybe not only of the editorial and since then, but just in general of your thoughts, your perspective on fMRI in general.
Well, thank you very much for the invitation to participate in this podcast. So I have to point out that I'm very much sort of going into the lion's den here as an outsider. Obviously, I fully understand the allure of fMRI. It's an amazing approach. You can do in vivo, non-invasive imaging of the entire brain and see it in action, and not just in experimental animals, but in humans. And I fully understand, with 65,000 papers, that it's accumulated an enormous amount of methodological refinements and so on. But so in my editorial, I have to admit, it was a rather provocative editorial, because for some time I'd been writing these rather anodyne editorials, in this issue, etc. I never knew if anyone was actually reading my editorial, so I thought I might reach into this cave and pull the tail of a sleeping beast, and indeed I think I did wake up this beast to some extent. So yes, I mean, I thought, one issue I had was this whole question of what the bold signal is. So it's as far as I can understand, and of course I'm hoping that you guys will correct me or educate me. It's a sluggish signal. It's non-linear. It's not suited to temporal deconvolution, or let alone spatial deconvolution. You can't really apply a sort of point spread function to try to understand what's actually happening at a higher resolution. And we don't really understand what it is. There's obviously this general assumption it's something to do with brain activity, but is it the activity of excitatory neurons, inhibitory neurons? Both? Is it synchrony? Is it the release of specific neuromodulators? All these factors are quite troublesome. And people of course will then say, well, isn't that the case for any signal that's reporting on brain activity, for instance, the EEG or local field potential or electrocorticogram? I dispute that. I think those are really much more directly related to neuronal activity. We can sort of understand the current flow, and the generative models for those electrical signals, I think, are a little bit more clear. And for instance, one can discriminate between synaptic currents, which generate relatively low frequency signals, and spikes, action potentials, which generate the high frequency signals. So in contrast with the bold signal, there really is, it's in a way so distorted to the point of losing the ability to really trace activity and processing all the way through to the fundamental signal that's being recorded. So that was really my concern about bold. Now, of course, I'm not going to, as I say, my editorial was rather provocative. I don't want to say that the whole field is flawed. I mean, on the contrary, one can still do an enormous amount and of course there have been some extraordinary discoveries and lots of success stories. But still, that was my starting point. So I don't know if you guys are going to now prove me completely wrong and say that actually the bold signal is extremely well understood, but I'd be curious to hear.
Yeah. Well, Vince, what are your initial thoughts?
Yeah, I mean, I would agree. I think in large part, we know that fMRI is indirect. I guess what I would say is important, though, is to kind of put it in the context of the strengths of fMRI as well, which are you get a very localized signal of hemodynamics, right? Whereas if you're measuring from something like EEG, and it's also non-invasive. So if you measure something from EEG and you're doing it like at the scalp level, right? You do have an inverse model. So you have a lot of assumptions that are sort of embedded there to try to localize, spatially the signal. You do have obviously a nice generative model of the current, the electrical signal from EEG. But it's complementary, right, in many ways, and that's one of the things that I do a lot of is basically kind of making the point that there's really no methodology that captures everything we might be interested in, right? So this is kind of in a general sense. They're all just snapshots, little windows, pieces of the elephant, so to speak. The blind men kind of feeling different parts of the elephant. But that being said, I think there has been, and Peter could probably comment on this a little bit too, but there's been a lot of advances in trying to, because the field's kind of recognized that we want to know more than just hemodynamics, right? We want to know something about, we can already do, sort of separate the signal components into things, related to blood oxygenation, blood flow, blood volume, and there's also work combining fMRI with other modalities, looking at which components of the signal is generating a functional signal. And there were some really interesting, and I'm forgetting the actual paper, but there were some really neat movies of some mouse data where they were showing kind of the signal flow from electrical activity through to hemodynamics, and it was a very interesting and localized mapping between them. Not to mention, there's some studies of looking at in 7T data, looking at layers, right? So input and output layers. Again, it's still kind of at the threshold of getting at details, but it is starting to tease apart again some of these questions of what is this signal telling us about, and how does it map into what we already know about the brain. And I think the other thing, which maybe we can get into later, is, and you alluded to this as well, Dimitri, is the fact that it's sort of a systems level view, right? So you're getting kind of the perspective of the whole brain and how it functions, which is very, has a lot of potential, right? In terms of understanding different aspects of, brain health and brain disorder and brain disease. But I'll just caveat that with sort of one of the challenges is of course, teasing apart which effects are sort of, proximal to the disease or disorder versus distal, right? So is it an indirect effect or a more direct effect? And there are studies ongoing to try to look at that sort of thing, but that's an issue. And I would say that's also an issue with a lot of other techniques. that when we're looking at function in particular of the brain. So
yeah,
I'll pause there. Yeah. So.
That's great. That's a good. And also just the way I look at fMRI in some sense is, I mean, it started out with some amazing effect. I mean, it just worked. So like way back in 1991 with our first experiment, you tap your fingers, you every single time you see motor cortex activation, and you see a certain percent signal change that occurs almost in every single subject every single time.
You don't need 100,000 subjects for that one.
Right. And so I think the way it evolved is that people were amazed that it just worked in terms of at that spatial scale, at that temporal scale, at that certainty, with every subject, primary visual, primary motor, it worked, auditory, whatever. And then they started doing cognitive functions, language, mapping, and so on. And so it sort of ratcheted forward. Everyone was sort of like just simply saying, wow, this works really well. And now we're asking further and further questions. And then obviously 1996 came along, and you had resting state connectivity that sort of simmered for about 10 years and finally had this breakthrough due to maybe more sophisticated processing or of being able to say, look, now with resting state, you see these connected regions that correspond almost exactly with the areas that we've already mapped out, and that seem to show this regular pattern across the whole brain. So even though it's indirect, and we know that there's many caveats, as you mentioned, nonlinearities, it's that the results showed a consistency and a segregation that seemed to correspond to what we understand about the brain already. So it still didn't really contribute much. And then of course, going to higher resolution and all the way along, I believe that what we're doing is what I think, and when I say we, I mean mostly maybe there's 100, hardcore methodologists who sort of are pushing the field with, comparing with other modalities and things like that. And they're trying to sort of say, okay, where is it, like you said, where is it nonlinear? Where is it linear? To what degree can we deconvolve the signal, and where does it break down? And now you're getting to people doing decoding, and decoding is also sort of a trick. It's sort of a proof of concept, and it has many advantages of being able to say, look, if we train up this algorithm, we can actually start to break apart, what a person is experiencing or thinking or whatever. And so it's sort of all indirect ways of getting at the validity of the technique and trying to push its applications. And yeah, definitely, as Vince alluded to, with higher, and also we've been struggling against things like large vein effects, for the most part, or draining vein effects. It's a hemodynamic response that limits our spatial and temporal resolution. But over the years, with either modulating your pulse sequence to be more specific to capillaries, or going to higher resolution and identifying veins in other ways, where it really makes a difference, where it really does have a negative impact, is when you really push the resolution to layers. And so you have to really be sure that you're not looking at veins. And so there's other technique looking at blood volume, in which we can actually get at layers. And then from layers, so there's been many false starts. Like for instance, and I'll stop in a second, people have been always trying to do like what's called Grainger causality analysis, sort of like using the latencies to infer directionality. And that doesn't usually work because of this. The hemodynamic response is so messy. But there's other techniques that are pretty much on the horizon. That is, if you get the high enough resolution to get layers, you can start to get upper and lower layer activity, and then start to infer output and input activation at the circuit level. And so that's kind of where we're at right now, where it's not quite there, but it's getting there pretty quickly at whole brain. So the philosophy has been, let's push the methods. Let's really try to understand how it does relate to electrophysiology. Let's try to figure out clinical applications. And part of the problem is that it works so well. There's a lot of people doing it, and there's a lot of false starts all over the place. And there's a lot of bad studies. And that exists because it's so easy to generate these maps. But there's a lot of serious scientists trying to model the signal, model the network effect, and really making some traction. So, yeah.
So can I jump in there
Sure.
and sort of try to push you a little bit on some of the other assumptions underneath, underlying fMRI. So, you point out that you get these big signals when you wiggle your finger, when you have a visual stimulus, and so on. And indeed, if I understand correctly, the strongest signals are in the primary motor cortex and primary sensory cortices. And of course, that makes sense. That's where you have sort of a maps. You have clear maps of the sensory space, or maps of the motor homunculus. But can one make that? I mean, what's the justification for the assumption that there is a similar sort of modularity where contiguous neurons or contiguous sort of microscopic areas are doing operations in blocks? So, for instance, the hippocampus, we know that place cells coding for contiguous areas in space are not clustered. They're distributed across the entire septotemporal axis of the hippocampus. So that sort of central assumption that you have a sort of modular block of tissue generating or underlying a computation sort of rather breaks down as soon as you get out of the primary sensory and motor areas.
Well, I would say that it still seems to hold in, people have mapped, have done parcellation, for instance, based on resting state of the entire brain. And so it seems to hold at the spatial. I think it's more a level of spatial scale than system. I think that, there's dorsolateral prefrontal cortex. It's clearly activated during working memory, but you're absolutely right. As you get in certain systems, especially subcortical, or maybe other systems, at the higher and higher resolution you go, the more heterogeneity you get in your distribution of your neuronal firing. And yes, it below about on the order of a millimeter, it gets hard to delineate easily the function. And yeah, our big assumption is that if you have dorsolateral prefrontal cortex, all the neurons are firing in a certain way. Even going to layer fMRI, we did a recent study with dorsolateral prefrontal cortex at layer fMRI, and you can differentiate the layer activity of working memory versus executing a task related to working memory. And so there's differentiation. So the higher resolution you go, the more differentiation you get. And yeah, there are very clear limits in which, and I don't think that, and I really don't think that in the studies, there's an assumption that everything is homogeneous. There's just simply an observation that it looks like it's consistent at this spatial scale. But to be able to make a statement about like place cells, I don't know if we'll ever get at place cells with fMRI. And that's an interesting question. So if we can't get to place cells, it is limited, but does it limit it in terms of its clinical efficacy in some ways, or is it limited in our deriving principles of brain function?
Yeah. I mean, I would say that yeah. So I think, Dimitri, you're kind of talking about, okay, yes, we can clearly get parcellation, functional parcellation with fMRI. We get these very reliable, maps, and this includes, higher cortical areas. It includes, these sort of functional networks that we find, right? And so basically what that is, it's just fMRI signal that's coherent, right? That's what we're inferring from. And so then I think the real question, in terms of getting at, is what's underneath, right, at a different scale or with a different, view of the brain. And so this is where we have a challenge, because it would be really awesome to have a way to sample at a fine-grained level from an individual brain while doing fMRI, or getting at some of those fine-grained level information. I mean, we've, for example, looked a little bit at once you have that data, if you had the data, right, you could start to do that mapping, and you can do that in some very limited ways right now. So for example, if you have EEG signal, from either an epilepsy sort of a brain EEG or scalp-based EEG, and you have fMRI, you can start to look at what networks that we see in fMRI are reflected in certain information that we can measure with EEG, right? And how does that tell us? What does that tell us? About those networks that we're observing. And so there's a lot of work left to be done there, I think, because but I think that there's also a lot of potential, because the maps themselves are, you can get them all, you can get them in one subject, right? Very easily, as Peter said, even, for these resting connectivity studies or other kinds of studies, it's very, very robust, right? And so, and it's not reflective of simple macroscopic anatomy. There's something, it's modulating with function, it's modulating with task, et cetera. So, I think one of the challenges we have here is that fMRI is trying to do a lot of things with a lot of unknowns all at the same time. So it's a macro level, right? So we're not at cells anywhere close to cells, right? So the early joke was it's kind of like putting your hand on the hood of a car and measuring the temperature and trying to say something about the engine in the car, right? We also have a lot of technical developments in terms of the methodology to collect the scans. I mean, Peter remembers the early days of trying to get fMRI scanners to do fMRI, right? That was really hard early on, and now it's kind of caught up, but there's still more coming. And then we have all of the sort of algorithmic side, right? So it's this very complex spatial temporal signal, and there's a lot of pieces in there, and we're still figuring out, which pieces are relevant, which pieces are not relevant, and under which context, those are useful. And so there's a lot of stuff to sort out here, right? So I would grant you that, I think, in the context of how do, we still have a lot to learn, right, in this field.
Yeah, of course. I mean, I'm fully aware of the additional information that fMRI brings to understanding how brain areas can be reconfigured. And in fact, that's an interest I've had, starting with had a PhD student called Thomas Acom who started this work in my laboratory, and we published a number of papers on oscillatory coherence. But it was very much a bottom-up approach of asking how could this challenge be met by, how one could reconfigure connectivity. We have a structural connectome, but then you have to have the ability to reconfigure a functional connectome, and we approached it from a bottom-up perspective. But of course, that always still makes assumptions of modules, and that sort of, I think I was listening to Daniel Bassett, I think she was speaking recently, and she made this interesting point about the sort of network approach not necessarily being suited to some processes, such as, I can't remember if it's ocean currents or meteorological phenomena. And I thought that's a very interesting point. If you how justified is it to go from the sort of modular approach? And of course, we have 150 years of lesion studies mapping function onto brain areas, but how appropriate is it to take the assumption that a sort of an area of bold signal that we know correlates to some area of a modular area of sensory or motor cortex has the same meaning when you go into higher order, sort of association areas, that it has the same sort of meaning in terms of a module of some sort of process, some sort of computation that is performed by that module. It's a reasonable assumption. I mean, it seems a very, very compelling assumption, but I just wonder if there's a ground truth. Is it a falsifiable assumption? If it's not falsifiable, then you sort of ask yourself, where are we? Are we on thin ice?
Well, I believe it's testable. I mean, it's testable by behavior and testable by, triangulating across other modalities, I guess, and also in animal studies as well. Yeah, I think that, and that's sort of what Vince was saying. It's like what we're doing is sort of pushing the technology and trying to test it at the same time in some regard. But I'm not sure exactly Right. I mean, so there's been a lot of work. I mean, most of the work now in fMRI is beyond the primary motor and visual and auditory cortex. It's definitely trying to look at um, uh, connectivity changes and meaningful differences in connectivity changes in prefrontal parietal regions to, you could have a whole laundry list of cognitive functions that they've been associated with. And it's repeatable enough that they can associate these things at a group level and to some degree individual level. That's what we're going towards. Yeah, so I don't know if that addressed, I'm not sure of your source of uncertainty as to why, and yes, there's certainly heterogeneity within those regions. And it just brought to mind really, really quickly, a nice sort of approach that caught on and maybe died down recently, Kalanick Grill-Spector, looking at fMRI adaptation. So you're looking at large areas, parcels that are several millimeters or centimeter large. And what you do is you give a certain stimulus that causes neurons within that area to habituate. And so only a fraction of the neurons habituate. Other ones still keep on firing just as they want. And then you can actually look at the modulation of the fMRI signal to see the modulation of that subpopulation of neurons, which is a nice way of kind of not really getting the higher spatial resolution, but probing at a higher spatial resolution this heterogeneity of neurons. So that there's all the field is filled with sort of clever approaches like that sort of try to get at the underlying neuron activity without making too many assumptions.
Yeah, that is interesting. But can I just come back to something you mentioned just a moment ago? I think you said something about areas of prefrontal cortex having different functions ascribed to them. I mean, am I not right to think that sometimes there's an embarrassingly large number of functions ascribed to the same area, and that's still a sort of it's a sort of a boxology approach where you have an area doing a function, whereas of course there's the null hypothesis that the function is performed by a distributed network that may span many areas of the brain, and so it may be a quixotic journey to try to ascribe a function to an area. But anyway, that's a very ancient debate.
And I think, what was it, a couple years ago, it was like cingulate gate, right? So there was this all these discussions about the cingulate cortex and what it does, right? And it was basically doing everything, right? So I think you have to be careful. There's the concept of reverse inference, which can happen in studies where you basically say, there's another study and they found this function was linked with that area, and I have that area in my study, therefore that area is doing that function in my study. But that obviously wasn't directly tested. That's just kind of making a loose association, not really a scientific, it's not really scientific evidence per se, directly.
That's a great example of bad studies in fMRI. I mean, Russ Poldrack sort of called that out several years ago, that there was starting to be a growth of those studies, and he's like, no, you can't make that reverse inference. But
right. I would say also, I think in terms of just network approaches, I mean, I think the other thing that I think has been a reason for why fMRI's kind of started to take off, even continue to grow in the connectivity domain, is because, the difference between fMRI, and I think we can it's informative to compare it to other modalities that have gained more traction in the clinical space, right? So structural MRI, looking at tumors, or CT, right? So CT has a real world unit, right? And fMRI, it's kind of a relative measure, and it's not just the MRI signal, which is obviously a weighted signal of multiple things, but it's a difference between those weighted signals. So it's 2 degrees kind of or more removed. But at least with like functional connectivity studies, and you can take issue with the word connectivity. It's really coherence. You at least have a normalized signal, right? So you're getting at something that you can start to compare, and you can scale it up and start to look at larger studies. So that's, I think, been really a useful and informative benefit. And I think the other piece is that I totally agree that like network-based approaches and module-based approaches are not necessarily always informative. And we've been looking, for example, at what we sort of frame it as sort of network aware and network naive, kinds of approaches where we sort of look at the entire brain in more of a, like looking at waves, looking at, different, kinds of approaches to it. So it's not, sort of a unitary approach. There's a whole bunch of different approaches, and some are better than others, and some are better in certain contexts than others. And I do think the field in general has a hard time making those distinctions, I would say. And so things kind of get grouped together, and there's another paper that comes out and another paper that comes out on these things. And it's, in some cases, it isn't particularly well thought out, right? In terms of why this, what's the rationale, so.
Yeah. Yeah.
Can I sort of jump into, I think but I'm not sure if we're going to advance the conversation a bit sort of too rapidly, but the sort of dynamic functional connectivity, the sort of changes in configurations, typically in these sort of resting state protocols, absolutely fascinating. And a lot of these changes take place sort of pretty close to the sort of the Nyquist frequency for the acquisition, if you see what I mean. You have a sort of, they're taking place at a frequency of about 0.1 hertz. And that's roughly where your cutoff is, because you can't really go much faster than that. So there's two ways of going forward. Either that is the relevant frequency at which these reconfigurations take place, and they have some relevance to conscious or subconscious processing, or some important physiological regulation, or you're just looking at the tail end of a hidden distribution where most of the work is actually taking place at a higher frequency, but it's invisible to your method. I'm just wondering how you guys feel about that.
Yeah. I mean, I think I guess there's different aspects to that question. One of which is the acquisition, the Nyquist frequency of the acquisition, right? So I think it's pretty clear that we are, you can sample now from the whole brain, at 300 milliseconds, right? Or 350 milliseconds or so. And so the Nyquist frequency kind of goes way up beyond 0.1 hertz. And you still see a lot of power at that frequency range. It's not only at 0.1 hertz. And one of my critiques of the resting fMRI field is that everybody filters out everything above 0.1, right? And there's a lot of information above that, right? And there's some studies that are coming out to kind of talk about this. And I think the other piece of this is that the hemodynamic response, is, if you model that as sort of a temporal point spread function, it's filtering the signal down, and it is kind of centering it in that range, right? Given what we know about, at least if we do basic experiments, the timing. But that being said, there's also it's not a monolithic point spread function, right? And there are higher frequency components to it that we can start to tease apart. And so I think I don't think that the 0.1 is an artifact of the acquisition at all. I think it could be related to the fact that we're sort of in this hemodynamic range. And I totally agree with fMRI, especially when you're doing whole brain fMRI, you're doing a lot of integration, right? So there is multiple communications that happen between these regions by the time you've gotten that signal, right? And then the hemodynamics have come down. So that's another piece where sort of trying to get at some more causal studies, or tying it to higher frequency electrophysiologic studies is needed to help us tease it apart. But it also doesn't mean that macro level signal doesn't contain a lot of useful information at that different scale, right? So again, looking at multiple scales is really useful and isn't done enough. And you can do that in fMRI. You can also do it in electrophys, and you can start to link, try to link those together. So.
Yeah, as a matter of fact, there's a really nice work. I think, well, Vince, you do that as well. But I just saw a recent paper a recent talk by Mark Woolrich at Cambridge, actually doing MEG and EEG with fMRI, and actually using hidden Markov modeling to sort of probe the dynamics and actually finding something we've always known, that the fMRI shows this slow changes in connectivity that's limited by the hemodynamic response. Yeah, I don't think it has anything to do with the acquisition limits. You can sample faster than the hemodynamic response. But what Mark showed is that if you do this with more electrophysiologic measures, yes, you get many more rapid transitions, but the pattern is pretty much the same, and the way the reconfigurations are similar to with fMRI. And so, yeah, I mean, fMRI, I'm sure there's a continuum of manifestation of power, and fMRI just sees a little bit of that, but enough. And we're probably missing a lot. But
yeah, I mean, I agree. I mean, we've done some studies, as you know, of like concurrent EEG fMRI, right, and looked at the dynamic connectivity configurations and the like, and it's pretty clear that there's a strong relationship there. But you can also play games again with a lot. You can do simulations, right? You can simulate these different scales, right, and you can start to see, I mean, things look quite different if you look at a different scale, right? And you're integrating over. So like you said, Dimitri, if you have this sparse spread, of firing neurons versus sort of a dense homogeneous region, but your spatial scale is broad and there's a lot of smoothing that's occurring, those are going to start to get blurred together, right? And it will look like a homogeneous. You're not going to be able to tease apart, the interdigitation or the like. So again, you're dealing with the data you have at the scale that you're looking at, or at the scale that you've chosen to analyze it at. And so that context is really important.
Yeah.
So we all agree it's a fascinating high-dimensional world, and there's this explosion of hypotheses that one can then probe. And as a rather old-fashioned sort of hypothesis-driven scientist, I sometimes despair at the conclusions I read of papers that are studying resting state fMRI, concluding that there's greater or lower betweenness centrality, or some change in anti-correlated connectivity between different networks, and so on. And I never know what's good, what should, in these groups of patients with a disease compared to controls, when they have one or other of these changes in metrics obtained from graph theory, what's good and what's bad. Sometimes it's a bit worrying because the next sentence concludes that this must be some compensatory effect. So I'm just wondering, what's the null hypothesis? What's the optimal level of between the centrality or eigenvector stability that we should all strive for?
Yeah, that's a really good question. Right now fMRI is sort of in the state of, one, we're sort of trying to figure out. I mean, right now the field is, there have been many clinical studies of populations, and you see population differences, and you're absolutely right. I mean, an example of not the best study is sort of like you compare different groups, you see a difference, and then you sort of have a just so story to sort of explain it. That's not as good as it can be. If you sort of try to triangulate with other methods or other, and you start with an hypothesis as to what should happen, that's much better. But still, the reality is that there is still enough variability in the connectivity patterns, and enough variability between subjects, between days, that we're finding, I think the field's at a state where we can't just do these N of 100 subject studies. We need, the Oxford, the database of thousands and thousands to try to pull out differences to potentially use as biomarkers. That may not be the answer either. So we're at this stage where we're trying to get a handle of the reproducibility of the networks, and we're trying to boost up the effect size. which would be better. And then, yeah.
Well, I mean, I would go further and say, I mean, I guess make the point that there's this sort of, again, it's a spectrum. There's a lot of spectra here, right? So there's a spectrum between group level studies and individualized studies, right? And it's sort of a cycle, right? Or a circle, right? And I think we need to go back and forth between them. And if we really are going to, if fMRI is going to make an impact clinically, it needs to be single, at the level of one individual subject, right? And there are a lot of studies, that have been showing useful and clinically relevant information in single subjects, right? So there's, but sometimes it's related to, I think there's some advancement now in the field in sort of trying to predict, for example, cognitive decline, right? In subsequent cognitive decline, or predict sort of biomarker validated decline, right? So you kind of have like a CSF marker, for example. Other studies have looked at sort of multivariate, kind of prediction of things like pain signatures, and the like. We've done a lot of work, and this is, I think there's also something to be said for recognizing the fact that we have, fMRI in a sense is kind of trying to chase its tail because it's sort of trying to be applied in many cases to disorders that don't have a mechanism underlying them. At least we don't know yet, right? Or they're very complex. There's a lot of studies. I mean, we do a lot of work with mental disorders and the like. In obviously neurology, neurological disorders and diseases, it becomes, you at least have something, to validate, right? To try to validate against. And so I think that's another important thing is that, you're trying to study something that isn't well defined in a lot of cases, and your ground truth isn't really a good ground truth. And so that's another piece that's kind of muddied the waters in terms of fMRI. But I think there's also been some advances there. So
yeah, and I think an important point here is that other fields, right? If you have mouse studies or animal studies, you can actually say, oh well, there's if you block the calcium channels, or if there's a mechanistic start, and then you can say, oh, this might be associated with this disorder, and you can actually draw the connection. With fMRI, you have activation patterns and networks not exactly lining up, and we don't know why, and we don't know how, and it's sort of there's a limit to the depth in which we can interpret fundamentally what it means. And that's why work of Danny Bassett or modeling the fMRI response, trying to make a mechanistic explanation at that level is also useful. I mean, I push people all the time, that you have to go beyond having fMRI as just cartography and get into more mechanisms, but also at the same time, I think that we don't know, right? I mean, we don't know if we're trying to derive principles of brain function. There's probably principles at all different scales, and fMRI, there might be principles to be derived at that scale as well. I also like to bring we haven't yet come up with also the clinical manifestations of disorders might be at the scale of the fMRI signal as opposed to the single neuron scale. I don't know that, but there might be different disorders for whichever. So that's why you need them all in some sense.
Yeah. Oh yes. I mean, I think the disorders of brain communication, I mean, one can call them functional disorders, except in neurology, functional disorders has a different meaning. But anyway, we call them functional disorders in the sense that they are disorders not of the fundamental structure, or there may be some subtle structural alterations, but the disorders where it's the abnormality of the information transmission or signal transmission through the brain, which is at the core of the disease. I think those are disorders for which fMRI, of course, holds out the greatest promise, except, of course, sometimes we don't have a ground truth. We don't have a way of validating this. Epilepsy, I sort of think, is in a sweet spot because, of course, we've got the EEG, and we know that epilepsy, of course, epilepsy is a collection of diseases. We know that in epilepsy, you haven't got very big lesions, structural lesions, but these are abnormalities of flow of signals through the brain. I suppose you could argue that, because we have the luxury of the EEG and electrocorticogram and depth EEG and intracranial, other types of intracranial recordings, we know an enormous amount about seizures. And so the added value of fMRI has been pretty modest. We haven't really learned very much about how seizures happen from fMRI. fMRI, of course, has a role in pre-surgical planning because of the ability to localize motor strip and lateralize language and so on. And then I suppose, even one step closer to the hemodynamic response, migraine, where you've got people with aura, you have this massive neurovascular phenomenon that you can pick up with fMRI. That's in a way in a perfect spot, because it's we think that the spreading depression probably is very, very closely related to the process that gives rise to the Bolt signal.
Right.
But then, of course, if you go into the other direction and go into something like schizophrenia, you end up with that sort of slightly nebulous area of not knowing how to test hypotheses. What is the ground truth? And of course, again, just as seizures can be generated from numerous different processes, the different components of schizophrenia, the sort of different first-rank symptoms, probably can arise through different mechanisms. And indeed, I'm not a psychiatrist, but a few patients with schizophrenia who have come across, every one of them is different, and so it's not a unitary disease. It's not a homogeneous disease that one can really approach with so much confidence that one's going to get an answer from the fMRI. But until we have alternative methods, I'm not sure that MEG is really going to be is going to displace fMRI. It's still generating an enormous amount of material to try to understand.
Yeah, I think there are some domains. I agree, but I think there are some domains which are a little better defined than at least trying to target, brain disorders, which would be looking at things like predicting response to medication, right? Sort of looking at longitudinal studies in cases where the medication will impact the prognosis, right, of an individual. Or trying to look at subtypes, right? Or even this is a little bit more challenging, but this is more of a research question. But trying to sort of see if we can use the brain data to help inform the nosological categories, right? So they're not based necessarily on anything biological, right? But they're useful. Right. So to what degree do those things, overlap and are convergent? And so I think there's a lot of interesting work ongoing in that space. And I think that there are a number of examples where fMRI looks to be promising for sort of prediction, type studies. And so that's an easier question, right? I'm taking a bunch of data, I'm throwing it in, and I know what I want to predict, right? So you have an answer. It's clear what it is. It's not based on diagnostic categories necessarily that are poorly defined. And you can and if the prediction accuracy is high enough and it has a high enough impact on these individual patients, then that can be I think there's going to be some of those types of studies that will emerge as well.
Yeah, and
but there's a lot going on. Yeah. So I
just want to bring out one quick point too, is that part of the reason why potentially we don't have clear correspondence to, let's say, a diagnostic criteria of schizophrenia or whatever, is just because of the fact that I think that what we're finding is that we need to potentially guide those criteria with the brain imaging results. I mean, it seems like that might help sort the diagnosis, looking at the brain imaging results themselves. So there might be different types of schizophrenia. There might be different types of depression that involve disordered networks in different ways. And right now we just lump them into a diagnostic criteria, and hope that they settle in. But if they're not, I think that's not necessarily. It's too premature to jump to saying there's something wrong with fMRI, but more potentially that the story is more complicated than we thought, and that if we try to sort these groups based on how they sort of sort out, and then we might be able to come up with different differential diagnoses in that sense. That's the whole RDoC push at the NIH, at least, to try to have these more brain measures of these psychiatric disorders.
Yes, I mean, fMRI is a sort of endophenotype, or I mean, because the whole biomarker field requires a lot of painstaking replication in real world scenarios, not just the sort of the people who volunteer to be scanned, but actually going out there in primary care settings and recruiting patients, and then seeing how these biomarkers pan out, and also, of course, then demonstrating clinical utility. Is there evidence that if you stratify or then correlate certain outcomes or certain results to response to various treatments, that it's actually going to have an impact, and then can just be justified as an additional step? I mean, it'd be fantastic if one could sort of reach for the request form and just say, please do an fMRI on this patient. We're still some way off. I mean, we can do that for very, very narrow indications, but it's still not just around the corner. Yeah,
I completely agree. Yeah, there's a long way to go, and honestly, it's not clear. I mean, certainly fMRI has lent itself a little bit more to neurologic disorders that are a little bit more tractable, and psychiatric disorders, that's definitely not low-hanging fruit, trying to get at biomarkers related to psychiatric disorders.
Yeah, and I mean, I think I would say that's not a problem with fMRI. That's a problem with the categorization, how in studying disorder. So that's kind of a category where I believe fMRI can be really useful and informative, right? But
yes,
that's
but as a neurologist, I don't, I can't see that many places where fMRI is a validated tool to help with diagnosis, prognosis, stratification, or navigation for targeted interventions. I mean, there are a few indications, but it hasn't sort of panned out, and I can't see it taking over.
Well, there's one nice example. I interviewed Michael Fox, who does, basically looking at lesions and looking at neurologic or even psychiatric disorders, and finding, the lesions were all over the place, and trying to find some sort of unity to this. And he started to look at fMRI functional networks, and to the extent the lesions lie on these networks, the diagnoses are similar in that regard. So it's a way of potentially unifying lesion studies in that regards, to put them into a framework of networks. That's more to do with mechanisms, which of course are fascinating. And indeed, there was a very interesting paper from Bill Seeley and his colleagues at UCSF a few years ago, but looking at different neurodegenerative conditions and arguing that the connectome, the functional connectome, gives some prediction as to how the different networks would be affected, and how disease progression happens in these different diagnostic categories. So that's fascinating from a point of view of mechanisms, but as far as clinical utility, so when will we get to the stage where at the individual patient level, it will be useful to get resting state functional connectivity measures or whatever, is difficult to see.
Yeah, and I think obviously you'd already mentioned the sort of pre-surgical mapping one, but there's also some like functional connectivity studies of sort of tumor aggression and impact on other behavioral metrics. But I guess I'll just kind of identify what I think is sort of another challenge to the field, which is that, so because fMRI does require a lot of, sort of domain knowledge, right, to work with it. There's a lot of choices to be made. There's a lot of methods, to choose from. There's a bit of a gap between clinically relevant questions being asked and people that do the analysis, right? And so those worlds only overlap a little bit. And so I kind of feel, like it's not that fMRI has tried and failed in a lot of cases. It's that it's never been tried on it. Like, I mean, if you do another study of X versus Y, in a poorly defined space, it's okay, you got a result, but it's not going to have an impact because it's not a relevant clinical question, right? And so that I think would be, there needs to be more studies that are targeted at asking questions that are going to have relevance clinically, and I think there's a paucity of that at the moment. There's some, but there's not a lot. So, but there's a lot more that could be.
Yeah. Now, obviously, I haven't read all 65,000 papers on fMRI, but from my myopic perspective as editor of Brain until the end of last year, there was a very interesting paper which sort of questions the utility of fMRI in the sense that this is a paper by Salvalaggio et al., Michael T. Beldeschotten, and we're also authors on that, and compared the utility of the functional connectome and the structural connectome in predicting the deficits following strokes. And I think they concluded, if I understand correctly, that the functional connectome wasn't particularly useful. I mean, for functional connectivity in an individual patient was useful in predicting the outcome, the deficit, but of course that's obvious. But they argued that the structural connectome outperformed the functional connectome. I mean, I think there've been a few sort of letters to Brain since then, but it's slightly challenged that assumption. Of course, this is just one paper, and I can see that stroke is a very messy field because lesions are never really quite so discrete. There are a lot of subcortical strokes, and lesion burdens often sort of not revealed by standard imaging. But anyway,
and I think also I guess I would agree. I think, and there may well be cases where fMRI doesn't perform as well, right? It's a technique, right, of many possible ones. But I also think in part, given all of the methodological choices and differences, I think they're, I think more recently we're starting to see studies where like interclass correlation coefficients and things like that are starting to go up, right? And that's largely in the context of it's not very good in task, unfortunately. And I think that's, task-based studies in part because of the fact that we don't have quantitative units on the signal. And it's not normalized, right, in any way necessarily. But I think that in other studies of like resting connectivity and the like, you are seeing, ICCs going up, at or above like 0.7 and higher, and that's getting kind of at least in the potential range, right, of being useful. And you're seeing, certain levels of predictive accuracy that are sort of getting high now. Again, if you're going to do this in I would argue it's going to be informative to look at large N studies, right? To kind of, to the degree that the models are complex and we want to train them, we want to make sure they work. But I think, again, anything we do, if it's going to be clinically relevant, has to work in one individual subject reliably and well. So if there's something, if we're looking at it the right way, that will fall out if it's there, right? So
yeah, we're working very hard on and actually the answer is not clear. We're working very hard on increasing the effect size so you don't need these massive N studies. And to increase the effect size, you have to increase the fidelity of the signal, but also maybe trying to parse what you're looking for a little bit better, group your subjects better. It's hard to say, but once again, it's not necessarily a criticism of the field. The field is actually, it's interesting. I mean, it continues to sort of progress forward, and as we go along, we sort of get a better idea of what its limits are. And we're also continuously surprised at kind of what it might inform, but it's not clear enough. And so, yeah, it's such a, it's a flashy technique. You have these brain maps, and because of that, there's a lot of room for bad studies. There's a lot of room for misinterpretation and doing crazy, not very good things with it. But at the same time, it's a measure that we're still trying to understand. Not only what it's related to, but how well and with what fidelity it correlates with disorders. And so, I mean, there's this recent paper that just came by my desk that I forwarded to you by Sanjay Kim showing, a really nice study showing, different activation with excitation versus inhibition, which is, classic sort of, like you see a difference, you don't exactly know why, you don't know exactly what's going on, but you see something. And so it's a super sensitive signal to things that might be there, but, and we have to sort of, obviously then do the groundwork to further establish that sort of things as a method. It's, yeah.
Mm-hmm, mm-hmm. And I guess one other comment too is that the dynamics are really interesting. I think I would agree with that, and it's one of the things we focused on a lot in my group. And but things are changing not only temporally but also spatially, right, over time. And so we have to kind of try to figure out how to model that well, right? I think there's been a lot of advancement, in particular in the past, 10 years or so in that space. You look at a lot of the studies of functional connectivity are sort of averaging the data together, but it's a functional signal, right? And so, in particular, if you're doing a resting study, it's a poorly defined task, right? And so it really makes much more sense to model that in a more dynamic way, so you can capture the transient, the transient information. So I think that's gonna be really informative as we go forward in terms of, again, potential metrics that can be evaluated in the context of clinical questions, right?
Yeah, I mean, I can see that sort of capturing the dynamics, a bit like understanding turbulent flow, is a scientific advance in its own right. But coming back to the individual patient or the individual subject, it would still be very important to have clear hypotheses. And I get the impression that from when you're dealing with such high dimensional data, pinning down the number of degrees of freedom is just extremely difficult. It'd be really nice if there was some sort of, some convention on this is it, and then possibly some sort of registered reports. Those papers where you say, this is my methodology, this is the hypothesis I'm going to test. You register it, you seal it like a clinical trial protocol, boom, and then you do the experiment, and then you publish it, and you don't change your hypothesis halfway through.
Yep.
Which I may be misrepresenting the field, but sometimes when I see those sort of slightly just so stories at the end of the abstract, I raise my hand.
Yeah. No, I agree. I think there's a lot of those out there, and it's made worse by the fact that there's so many choices to be made, right? And so p hacking, et cetera, right, is very easy, and it's easy to inadvertently do as well, right? As careful as you try to be, right? It can be, you could still find out later, oops, there was some circularity there that I didn't consider, right? And so I think those kinds of things, I tell this story a lot, that I see this quite a lot with my students, where they get started on fMRI, and then they'll come and they'll be really excited because they've got this really high predictive accuracy of something, right? And then, ask some questions, and they'll go back and continue to kind of analyze it. And then they'll come back later and they'll be like, I just lost everything. It's not doing it. It's not performing well because I was using this information to do the analysis. That's why it was so good, right? But I didn't know that, so I shouldn't have used it, right? So I think it's, it's one of the reasons, I think one of the things we're trying to encourage more of are obviously data sharing, but also competitions and things like that, right? So you define a question, you put some data up, and you define, again, like a registered report in a sense, right? You're defining a question ahead of time, and then everybody's kind of competing, right, to try to see how well they do, and then the evaluation's done on data that they never saw, right, at the end. And so that, I think, is one way to try to help get at generalizable results that will that will persist, right? As opposed to, again, you can have the best group in the world doing something, and they might get something that is a little bit more only in their hands going to work, right? And it's not because they're doing something really special. It's just because something had was a little bit circular that wasn't considered. it's easy. It's just really easy to do. So.
Yeah. So, and you're right. I mean, I think that the field right now, I mean, the statisticians, the people who are the data scientists are, I mean, they're sort of playing a better role in terms of playing a more significant role in terms of how we handle this, because it's very clear, just as Vince said, and you know, some extremely skilled experimenter who's not doing anything wrong, who's doing everything right, but it's hard to sometimes reproduce those results. You know, the signal is variable, the signal's rich, which on one hand makes it really difficult to properly interpret, but on the other hand, it's a huge amount of opportunity to sort of, for those people parsing it out a little bit further and who are a little bit more systematic. So, yeah, I would argue we're still trying to figure ourselves out. I mean, fMRI has a place, and as I said before, I mean, it's very easy to do just so studies, get them published. It's harder to get those published. The field's sort of ratcheting up a little bit, but at the same time, there are more hypothesis-driven studies. There's a little bit more sophistication in terms of how people go about asking these questions, a little bit more trying to do multimodal analysis, and really more of an eye on the ball of what the goal is, not just to make you know, nice maps of the brain and say, look, there's a nice map of the brain, but really either to understand the brain, you know, maybe try to derive principles of brain organization, at least at the scale we can, and two, you know, I think in the last maybe even 5 years, the whole idea of let's get clinical traction on this has just really started up. You know, really trying to, you know, not only develop biomarkers, but even the low-hanging fruit. What I look at as low-hanging fruit is, you know, from resting state, you can derive measures of neurovascular coupling or flow in the brain, just looking at the latency of the global signal and things like that might be useful clinically, maybe even more useful than coming up with biomarkers of schizophrenia or whatever. So, you know, it's after 30 years, we're still debating things like how do you best eliminate veins but there's still tons of progress. And I think it's made mostly through the multimodal integration, people stepping back and really assessing the statistics a little bit better. And also the creative people who actually look at other aspects of the signal, like the latency, the width, the undershoots, which all might contain useful physiologic information that we're still in the process of looking for.
Well, you say it's more difficult to publish, but Brain still welcomes, I just checked with the new editor, Masood Hussain, he's still going to welcome papers on fMRI as long as they are relevant to neurology or translational neuroscience. And in fact, it's interesting. I mean, I had a look at the rate at which we were publishing papers on fMRI, and it's about 30 a year in Brain, and almost none in the sort of classical competitor journals, Annals of Neurology, JAMA Neurology, Lancet Neurology, almost never published on fMRI. And I don't know if it's because Brain is slightly different, it's slightly more scientific than those other journals, which are a bit closer to the practice of neurology. And I had a quick look at the sort of citation rates, and they're about average. But you know, if you look at the citation of average papers in Brain, whether they have and compare them to papers that have fMRI as part of the methodology, they neither get cited more nor less than the overall average. So I think fMRI is going to continue being an important part of what Brain publishes, and so I'd encourage people to consider the journal.
Okay.
I would encourage researchers getting into the field to start to target clinically relevant questions and well-defined, you know, just survey what's out there, right? We need people to do more integrative type, you know, surveys of, okay, there's like these million things, right? These are the ones that look promising. Fix them. Let's test them, right? Because in some relevant questions, because I think you see so many papers out there. And so since I come from more of an engineering background, I probably see more of this than many, where, oh, it's a real cool mathematical approach, but there's no connection to an application of relevance at all, right? It's just like, oh, we did this and we threw this data at it and we got that, right? But the method's really cool. So we need more people, I think, that are really targeted at, there's enough out there now that we should be able to do this. And there aren't a ton of people that are really doing the work to push that forward, to get to a real biomarker. There's a bunch of things that have to be done, right? To get something validated, et cetera, or to see whether it's going to be relevant, in terms of try it out, right? With a clinical collaborator, who you can work with closely. So that would be my encouragement to the field.
Yeah, we're trying to pull ourselves up by our bootstraps a little bit more. And yeah. So is there anything, it's interesting. There's right now at this moment, there's a windstorm going out outside my house, and I'm worried about the power going out. So I want to make sure this is recorded. But we've certainly gone longer than we typically go. And I'm certainly mindful of your time as well. Is there anything else that you wanted to make sure you emphasized, or anything to, there could be a round 2 of this as well. This is a great discussion that we should have more of as we go along. But is there anything today that you would be interested in just making a point of, or do you think that we've covered a lot of ground? But
I think we've covered a lot of ground. I mean, I've learned a lot. I'm as I say, I came into this a bit like a journalist, trying to understand the issues as an outsider, coming away with a greater understanding, a greater appreciation. It's been a learning process for me. I'm very glad that you invited me to participate in this. Yeah.
And I'd say as well, it's been really great to just hear your input and thought process, right? And it's very valuable. And I think this kind of a discussion is really useful. I think for me and for many in the field, to really just take a hard look at what are we doing, right? What are we doing that can be useful in these ways, and how do we think about that, right? So
yeah, I think this sort of discussion really is valuable, and having a perspective from those not sort of so much in the field who are outstanding scientists who are using other modalities outside the field. It's really valuable. Not only for us to evaluate ourselves, but for potential opportunities for, taking a different perspective and taking different directions potentially. So, yeah, we're not always aware of what our gaps are, and this is great. So thank you very much. And actually, thanks for writing the editorial that spurred this whole thing.
At least the 2 of us read it, right? So
yeah, the 2 of us read it. And then we read it, and we're like, then obviously other people became aware of it, and I think the value of it is definitely in the conversation that it catalyzed among a lot of people, and hopefully it will result in more. So,
well, thank you. Yes, I was slightly worried that I'd be the little boy that says the emperor has no clothes, but no, on the contrary, the emperor has beautiful, colorful, perhaps too colorful clothes. But we still have to make sure that the clothes are performing.
He's overdressed.
Function.
Yeah.
Are they keeping the emperor warm?
Yeah, exactly. That they do their job. Yeah. All right. Well, thank you very much. And
well, thank you.
Really do appreciate it.
Thanks, Peter. Yeah.
Thanks.